So so you cannot conclude that one to be a possible heat. It's not that it is structurally complementarity is enough for the docking. So there are so many things are needed. So what are the things we need to have? First, we need to have a protein structure. So where we need to look for the cavities in big enough to hold the ligand. So it's not the structural complementarity, but it should have a cavity to hold the ligand. So that is very important. So the first step is you need to find the active site of the protein. There are different active sites. So how you can identify active sites? So you need to find whether the ligand fitting cavity is there or not. That is the first thing. And then fit ligand in different orientation. So if you have a cavity, you find a cavity which can hold the ligand size. Then the next thing is you go and make the ligand to bind in that area in different conformations. You need to give a freedom to the ligand to fold in different ways, because it can go and sit there. The pocket is there, but in which direction, in which way it is better, that is going to be understand. So we need to give a freedom to the ligand to change its conformation and go and bind to the active site. Okay. So who will do that? You are not actually, you are not doing it physically. The computer need to do. Computer need to do mean there should be some program which can direct it, right? So that required different kinds of algorithms. So that is, we are going to understand different algorithms which are there for this kind of docking. Okay. And we also try to understand different software, actually, how to identify the pocket or the cavities on the protein, so which can fit the ligand, which could be a possible active sites. Okay. Okay, it's go and bind it. So bind, how you can say that it is a binding? It's not a physical phenomena, right? So it's not like structurally just complementarity and physically fitting is not enough. So it should have some interactions. There are so many kinds of interactions as we discussed. So it can be hydrogen bonds, van der Waals interaction, hydrophobic interactions, all those things. So that makes the ligand to bind over there strong enough. Okay, so that one, how we identify? Again, we are not doing anything in vitro conditions, how strong it is, how stringent uh, no procedure it's required to detach. So if you do in vitro, then we can understand that it is binding. So we are doing in silico. How we can do it? We cannot just say that it is go and attach it. If you put any two molecules beside, it looks like attached. How you can identify? We need to, the third one is very important, that calculate the binding energy for the different orientation and conformations to identify which orientation is the best. So when you are allowing the protein and conform the ligand to different conformants binding at the cavity, we need to calculate the interactions and give some score. That is very important. So giving the score is important. How you identify which conformation is better? So last week we have done the auto docking. So what we have done, the protein target we have taken, one ligand we have taken, and asked them to go for different conformations to go and bind at the active site. So we already shown that active site where it is. And even though we show the active site where it is, and how the program can understand which conformation is better, which pose is better, because whenever it is go and attached to that area, we are calculating, the software is calculating the energy how they're calculating the energy. They are calculating, it is calculating the possible bonds it is forming between the ligand and protein. So that is the information we have to give. Who will give? There are some softwares which already they have designed for it. So our task is just to use it only. We are not identifying, we are not no developing any software for it. So just what we are doing, we are utilizing it, and we have to understand. So what is happening behind it? Though we are using, what is happening when the ligand and protein is coming together, and it is the program trying to understand what kind of bonds will form between it, and it will give some score for each kind of bonding, and it will give a total score of that particular interaction. So then it will go in it will remove it, and it will go and another confirmation, it will try to bind it, or orientation. Then what is the interaction? Then what is the score? So taking all the scores, and which is the lowest free energy. So here the score has low, it is better, that is going to be the best uh, best interaction. So as we can see in the universe, whichever when you have the lowest free energy, any of the molecule, so that is going to be um, more stable structure. So thus, so we need to understand which conformation, which interaction is having the lowest free energy, so that we can take that as the the best conformation of the ligand, and that is the best orientation of the ligand to have the interaction with the drug, sorry, the interaction with the protein or the target. So that is the main the output of this docking process. So for that, so these three things we need. First, we need to have, once we have a protein, we need to find the active site. So then once you find the active site, okay, we can tell the program to our computer that, okay, this is the active site. No need to go everywhere on the protein. Just put the active site. So here you go and have an interaction with the ligand. And when it is, we are introducing the ligand, we need to use a different way of um, giving a freedom to the ligand, different orientations. When it's good, different orientation one each time, and the each orientation should have a score of after the interaction. So then after few orientations and confirmations, and calculating each interaction, so it will give a score for all those. So we go with the tens of thousands of interactions, but on the day we have not done that much. I think we have done, I don't remember, maybe 100 or something we have done, or maybe 1,000, less than 1,000. But that is just for the practical purpose. But if you want to go for really identification, we need to go for tens of thousands of confirmations, sorry, orientations we need to give, confirmations we need to give freedom. So it will find the best one. Okay. So then that is another software we need. Okay. And the last one is calculating. So what kind of calculation? There are so many different kinds of calculations, because these are all man-made, right? So it's not that which is exactly true, we don't know. We are just predicting what could be the interaction. So if it is that interaction, okay, which is the better, which is the most uh, strong bond, and what is a weak bond, what kind of interaction we can give a better score. So all these kind of scores, each scientist come up with the different kinds of calculations, and come up with a different kind of algorithms. So there are a few algorithms are there. So that is what we are going to understand. The first one, finding the cavities on the protein. Okay. So if you want to have a active sites identification on the protein or the target molecule, before that, first we need is structure of the protein. Where do we get the structure of the protein? So two ways, experimental or computational. If you have already the structure known available, so most of the times it is a X-ray crystallography structures or few times NMR. If both are not available, very, very rare the electron microscope. So, and you can go for homology modeling as last week you heard. So at the beginning, if you don't have exactly structure of your target protein, like spike protein, when they wanted to target it, when human new coronavirus spike protein was not crystallized, not studied its crystal structure, first it has taken the structure of the um, source one. So that one, is a it may not be exactly similar, but based on that the structure and based on the structure of the COVID-2, and both align it, and wherever it is having a different sequence, so we can take the help of homology modeling, and then we can predict the structure of it. So thus, the structure of the protein will be ready. So once the structure of the protein is ready, then we go for the active site or binding site search. So how we can understand the active site? So last the practicals, what we have done, active site, how we understood? We understood because we have a structure, a crystal structure of the protein and the ligand together. So it's already there. So then we can understand where is the active site. So that is the literature based. So there are two ways, literature based or computational algorithm to predict the binding site. So literature based mean when you have already a crystal structure where it is having a together protein and ligand together. That's what last week we had. The structure we have downloaded from the BDB was, it is not only the protein with a ligand together, the structure we have downloaded. So that once you know where it is, the ligand is binding, so that is the active site. Okay, so we no need to search for active site separately, because we just put that is the middle of the one of the amino acid we can take with at the binding site, and the surrounding maybe we can give a 5 microns Armstrong or 10 Armstrong for the active site to go and bind by the ligands. So that is how we can understand the active site from the literature or from the crystal structure. Or sometimes if you don't have crystal structure, but some studies may have be available, like, okay, this amino acid deletion may be affecting the ligand binding, or this nucleotide structure mutation is leading to the weakening of the binding of the ligand interaction. So once we understand by mutational studies, so which mutation is leading to the poor binding, so we can predict what could be the active site, because that amino acid it could be a part of the active site. So we can take that as a middle, and surroundingly we can make a grids. So that is the we can define as a active, not define, is a possible active site, we can locate it. Okay, that is the literature based. If you don't have NMR or crystal structure, X-ray crystallography structure, or even not having any mutational studies, so how to go? Just to, it's a new protein, nothing known about it, and we just need to predict the active sites of it. So then there are some algorithms which can help you to predict the active site, like alpha site search, Lenhoff search, Canoli surface mapping, Coons et al. Clustered sphere search. So none of them are so great, and I mean, great in the sense perfect, and at the same time, they are not bad also. So it will give you some kind of information. But if you see Maybe that's how it's only a limitation, because we are just trying to predict the way how to find the active site. So one of the way is Connolly surface mapping. So this is a kind of protein. So rolling sphere model, it's called a solving accessible area. So 1.4 Armstrong, which is the water molecule size spheres, will be rolled on the surface of the protein and find the grooves on it. What are the grooves present on the protein? So who is be doing? Just we are simulating the structure. So on the structure, computationally we are rolling all the probe spheres of 1.4 Armstrong, so that we can understand what are the grooves are present. Otherwise, just the amino acids which are having the atoms, you cannot understand what is the structure of it. So this is the one way to understand the solvent accessible surface area. So to understand the surface of the protein. So this is the thing, it will get it. So something like the structure you will get. So then we can understand, okay, this is the groove, something is there, or three-dimensionally, there will be anywhere, grooves will be there. So possible ligand binding site. That is what, it may be not one, maybe a few, maybe 10 or 8 or 7, like that. Some of the ligand binding sites you can predict based on the surface area. So another one is Lenhoff surface, where the complementary surface for the ligand instead of Connolly surface of a receptor. So possible positions from the atom centers of the ligand. So it is taken as the atoms, this one. So it will be taken between the atoms, what are the edges are present, similar like this, and based on the contact score of uniformity distribution points on the probe force. So something like this, it will try to find the surface of it, and try to find the grooves on the the structure of the protein. Another one is Coons. Coons are it is going to do something like, it will first roll the big circles and try to understand whether the two corners of the circle is actually attaching to the protein or not. So when it is, you can see here, when it is going for uh, groove into the groove, so it will roll a smaller circles. So like that, when both ends need to be attached to the two different surfaces of the protein. So based on by rolling of these different sizes of the circles, and and matching with this distance between the two surfaces of the protein to the circles, so the software can understand where the grooves are present on the surface of the protein. So there are cavities on the protein. And alpha shapes is another one. So you can see the meaning of it. So it is it will take the atoms middle point of the each atom of the protein, and take alpha is the radius of the circle, and wherever it is mapping the two corners of the two atoms, so it will give a structure of it. So something, if you have a bigger alpha radius, then you will get a different structure. If you keep lowering, it will give a different. For example, see alpha, if you put a very big, so you will not define a proper structure. If you lower it, you can see, you can understand possible grooves or cavities of the structure. So that is what, see, if it is big, so just you will find this structure. If it is smaller, getting smaller, this structure, and but bit smaller, this you can see, and bit smaller you can see. So like that, okay. So whatever it is, the way they're trying to understand the possible structure of the protein, especially to identify the cavities present on the surface of the protein, so that when the cavity area, if it is matching, or at least if it is the similar or bigger than the ligand size, so you can dock and see. If the cavity size is much smaller than the ligand, so no point of uh, docking. So there is no, that we cannot consider as active sites. So this is how first we can predict the active sites if we don't have the literature information. Okay, so this is the, just to have an example how the alpha shape, what it will get, and the colony surface. So it will give a different shapes and different cavities. So here it looks like cavity. Here also it looks like cavity. And here small grooves is there, but here you don't have as much grooves. So it's have its own advantage and disadvantages. But anyhow, it will just predict the possible surface area and try to give a small information on the cavities present on the surface. So that is the first thing what I was trying to tell you. So this is the looking for the cavity big enough to hold the ligand. So this is just now we understand, if you have a literature information, we can get it done. That is much easier. And if you don't have, we need to use this software. So just need to download that and just give an input and you can get output. So it's much easier. It's a very, very easy because you are not developing any program. So if you want to develop a program, you need to understand the chemistry, you need to understand uh, the mathematics to find which is all the calculations. Then you need to understand the physics, you need to understand the computer knowledge, that means scripting knowledge, and the statistics also. So if you want to develop all, you need to have. But we are not developing as molecular modeling course is just you need to know what is going on background and how to use it. Okay. And then the second thing is fit the ligand into the pocket or the cavity. So there are two ways we can do that. One is rigid docking and flexible docking. Rigid docking is very simple and is not effective at all. Rigid is just keep the space, the shape of the protein uh, and ligand structure also unchanged. So just both conformations are defined, and just put this one and this one together and just check the calculate the interactions between that and find the energy. So you're defining already ligand how to bind and exactly where to bind, but you're not giving the ligand to change its different conformations, because ligand structure, we don't know exactly how it is. There were so many possible orientations. It can, maybe this structure may come here, this may go here. We don't know which is the best way. But if you define it already and just do the docking, that is called as rigid docking. Rigid docking is very simple. It will not take time and computationally very cheap. So it will not take much time. It's just few calculations only. And but the limitation is it's not much effective. It's just like a key, lock and key model, okay, but inaccurate. So the best way is flexible docking. See the ligand. So protein, you can keep defined structure. So intact, you can keep the protein structure intact, but you can take this ligand, see the ligand, and you can give a freedom to change its conformation. So it changed this one to here, and this one is changing to this, and this one to this. See, this one is going to find nicely fit into this binding site. If you put only this one, the first one, and without any freedom, you go and ask it to bind to the protein, it may not bind effectively. So you are end up with, you are not, you are losing a potential drug candidate. So that's why you need to give a freedom to the to the ligand to change its conformations and go for docking. So it is just to show you this complementarity structure. It's not, it will not work like that. We don't know exactly which shape is going to fit unless we just give a freedom and let it bind, just to make you understand. So just shown here. So this is the best to fit, but in fact, you cannot understand like that. So that's why you need to have a way how the ligands have different kinds of structures and go and bind to the protein. How you can check it? How we can, the computer can check different conformations to check with the protein's active site. So for that, different algorithms we do have. Okay, dimensionality. So we are giving you freedom at the position X, Y, Z. We have seen, right? Every atom, when you place that at the position, it is having its own X, Y, and Z what do you call, coordinates. So that is also we are giving a freedom. So three-dimensionally, all the coordinates you can give freedom, and then orientations. Just now you have seen the atom, each atom that A and B atom like this one time and like this one other time. So you are giving a orientation also, you are changing, and bonds also you can give a freedom, torsion angles you can give a freedom. So if you take all those total freedom, it's a lot. So you can give a freedom to the position, that is coordinates can change, and orientations can change, and torsion angles also can change. So torsion angles is based on how many atoms are present, how many bonds are present in the molecule. So all these different kinds of freedom. So if you want to give, there should be a systematic method how you can allow a different structure go and bind and calculate the score. So that's why there are so many different kinds of algorithms were developed. One of the way is, there are three different ways for this flexible docking. They are called as stochastic method, systematic method, simulation method. Okay, stochastic method is it follows the genetic algorithm. Systematic method, it follows the incremental algorithm, and simulation method, it follows the grid-based docking. Okay, let us understand the three ways. Very simplest way, I'll try to explain to you. If you refer any textbooks or any review articles, it's very complicated, because all it required, you may enter into the statistics, mathematics, computational scripts, all those things. So without touching all those things, simplest way I'm trying to give to you, having an idea. So first one is stochastic, a random method. So it follows the genetic algorithm. Example is Gold, Autodoc. So Autodoc we have seen, right, last week. So Autodoc is a software where we do the docking, and Gold also another famous one. After the Autodoc, another famous one is Gold, also follows the stochastic method. What it does? So it uses the genetic algorithm. Using randomly generated population of conformers, evaluating the fit, cross over the retained fitting molecules, and then iterate. It's a very simple, take a molecule, ligand molecule, go and bind to the protein, active site, take the score. Score is another algorithm, so it will take a score, then come back, change the structure, and again do it like that. Around 10 it will do. Okay, around 10, let's say. So out of the 10, the best one will be kept aside. Okay, another random 10 it will do. Okay, the best of those 10 it will take, and then the next it comes this best and this best, and it will go hybrid. Okay, like just like your chromosome, you see the p arm and q arm. So half of it maybe in the first structure, and half of it the second structure. It will try to make it the hybrid and again search for it. So best of the best it will keep taking. So that is why it is a genetical mean what, that is what, right? So have a parents and the progeny. So similarly, so best among the random orientations are taken as a parents and hybrid them to find the which are, you know, the progeny molecules structures are better for binding to the protein active site. So that is how it will use the algorithm to generate the different conformations and find the best one. So otherwise what happens, there are so many of uh, possible the orientations. So it will, if you want to know everything, it may take a very long time and it may not be a proper way. So instead of giving the random freedom to for the orientation, a systematic way that take this and this and get the best and hybrid them and go for it, and then out of that you try to get the best, maybe then hybrid another two of them, and getting such a way. So it will get the best way, best one orientation, which can nicely fit into the active site of the protein. So the next one is systematic method. So systematic, the examples of the software using the systematic is a Flex and Glide. This software, they use the systematic. So systematic is like, say, this is the ligand structure. What it will do, it will split into a different parts. So it will split into, okay, this is the aromatic, this is the aliphatic or hydrophobic, this is the hydrogen bond, and something like that, ester bond. All this is split into it, and try to find which one is the best for which kind of interaction. So, okay, this is the aromatic, go and bind to the aromatic. And this is the aliphatic, okay, go and bind to the aliphatic. And this go ester bond or hydrogen bond, go. And after binding all these things, link each other by just linking the bond. So that is how it will predict the possible structure. So here, see, this is the structure is different. This is a structure is different. So it is a orientation. It is predicting which is the best way based on the the different uh, you know um, functional groups of the ligand and the protein active site. So that is the systematic. Then okay, this is the one. So the ligand is cut at rotatable bonds into fragments. Fast initial placement of the fragment is important, then energy optimization. So after that, how to make it into lowest energy possible to have more interactions. So this is how. Then simulation. Simulation is followed by glide, autodock, and dock. Okay, what it will do here, here the once you find the active site, you will be it will be given a space surrounding to it from a grid, a grid box, okay, which we have seen in the last practical also. Grid box, it will be formed. The grid box, right? So it will be, the grid box is not just a physical. So inside the grid box, it will be given a score for each corner, each point of the grid. Okay, so let's say this is the grid box. Okay, here the hydrogen bond possibility of the active site. So any molecule coming here, so give a score. Okay, any molecule coming to that point, give a score. What kind of molecule? If it is hydrogen or oxygen, is a hydrogen donor or hydrogen acceptor, so give some score. Other than that, give another score. So like that, every point of the grid will be given a score. So that is what happened when we made a grid in the before the docking step. Just one step before the docking, we made a grid. So what is the grid? Grid we are giving is, so this is the active site, and surrounding that you make a grid. So 10 Armstrong or 5 Armstrong grid. So that is a cube it will form. So inside the grid, every corner, every point, it will be having a certain score. So that will be pre-defined. That is what happens when we make a grid calculations. So that's why it is take more time for the grid than the docking. So that is what simulating. Everything is simulated inside the grid. So after that, once you enter your ligand inside, so it will direct which is the best one score, which orientation is the best. So that is the simulation. Okay, so that is the next one. The final one is the, so we have understand how different orientations enter into to dock to the protein. So then calculation. So just docking to the protein active site is not enough, because as I was stressing you that it's not structural complementarity. It is the interaction is important. So what kind of interaction? So what kind of score it need to be given? So there are different scoring functions are there by different people, and they are force field based, knowledge based, or empirical based. And force field based used by doc, autodoc, G-score. Knowledge based is drug score, PMF, and empirical by Glide and Ludi. Again, here, if you want to go and deep understanding, it required a lot of, your chemistry and mathematics. So what I do is superficially, I give you a rough idea what actually they are on, and how it work, how it is beneficial for our docking. So ligand and protein, when we dock each other, they bind each other. So ligand protein will be formed. Okay, with that, some of the water molecules will be removed, which are in touch with the protein and ligand before, and we are removing some of the water molecules from it. So by that, so there are some changes in the protein ligand interaction: hydrogen bonding, hydrophobic effect, electrostatic interaction, van der Waal interaction, strains in the ligand. So when it is binding, so there are some of the bonds are in the ligand maybe got got strained, and the angle may have a little bit stretched. The same thing might have happened in the protein also. It's not the structure, okay? It's just bond only. Between the two atoms, the bond may be stretched slightly. So that one we need to take into considerations for it. And yeah, force field is the affinities are estimated by summing the strength of intermolecular van der Waals and electrostatic interaction between the atoms of the two molecules in the complex. The intermolecular energies of the two binding partners are also frequently included. Finally, since the binding normally takes place in the presence of water, the dissolution energies of the ligand and protein are sometimes taken into account using implicit solvation methods. So there are different methods whether you want to consider the solvation energy or not. because some of the water molecules are removed from the surface of the different two molecules, and other some of the water molecules still present. So calculating all those things in using the force field also, there are different algorithms are there. So one is the charm, where is the total free energy calculated by some of the bonds, and this is the called as UB and angles, dihedrals, impropers, non-bonds. All these things will be considered, and then be something. Forgot something. Okay. Then this is the amber. Also, kind of the force field only. Also, it will consider all the bonds changes. This is called as changes in the energy due to the bonds and due to the angles or due to the changes in the dihedrals, non-bond interactions, hydrogen bond interactions. All will be taken into consideration and given some score. This score is important. That is how you can understand the lowest score is the best interaction. So empirical one actually counting the number of various types of interaction between the two binding partners, either based on the number of ligand or and receptor atoms in contact with each other, or by calculating the change in the solvation free surface area in the complex to uncomplexed ligand and protein. So solvation area mean the water exposing surface area of the protein and the ligand. What are the changes occurred? This is the delta is changes. Okay. The coefficients of the scoring functions are usually fit using multiple linear regression methods. These interaction terms, hydrophobic interactions, that is favorable, right? Hydrophobic always favorable. Hydrophilic unfavorable. Hydrophilic is a favorable. No contribution to affinity except the following special cases, like number of hydrogen bonds, number of hydrogen bond mismatches, all these if it is hydrophilic and hydrophilic. And number of rotatable bonds immobilized in the complex formation. So these are all the things it will take. So Gibbs free energy, and then changes due to the hydrogen bonds and metal interactions and rotatable bonds and etc. So that it will give you the change in the Gibbs free energy after the binding. So this is called as chem score. So similarly in empirical scoring also different algorithms are there, like a chem score, this is the glide score. So different, they each one come up with a different way of calculation. So we cannot exactly say which is a better. So you have to use, when you do the really in a research, you have to find different ones and find out which is the best one. Though you are not actually developing anything, just finding which is the best one. So the same process you can use using a different energy calculation algorithms and see whether you get a same best one or different. And if you have already like an interacted one, maybe you can check which is exactly showing the already known one. So you can predict that could be a best one to be used. So Autodoc 3.0 is another, its own empirical scoring method. So these are the calculations, how it works. And knowledge based. Knowledge based is the based on the knowledge, as it is mentioned. So based on the statistics observations of intermolecular close contacts in the large 3D databases. So we have to take into consideration, so how the 3D database we have ligand and protein interactions. So based on the interaction, based on the which kind of interactions will be better to be really to be a ligand binding. So those kind of interactions to be taken consideration and then give the kind of scoring. That is the knowledge based. So neuraminidase inhibitor, I think we have discussed about this in the last previous class. So this is one of the example of the model system study where they have come up with the drug neuraminidase, which is natural one is a dana, which is the natural ligand or a substrate. But similar to this structure, they found another one called as Relenza by GSK. So this is a potential drug to target the virus. So